All right, so we'll continue our journey on the immune system. So now we're going to look at the adaptive immune system compared to the innate immune response. So then we'll talk a little bit more about antibodies and antibody generation. And finally, we'll get into more of the signaling molecules that can mediate an immune response. And these signaling molecules are usually involved with inflammation. So we'll go to the inflammatory process next, and then we'll talk about some of the drugs that are used to combat inflammation. And then we'll go to some immune system degenerative disorders, such as arthritis. And we'll talk a little bit more about allergies and asthma as well. So all of these topics are related to immune system function. In talking about the adaptive immune response, we need to get just an overall feel for the anatomy of our lymphoid system. And so we've already taken a look at the lymph system, and this is a very cool page on this website. So this is also the Stanford website that we've been using. So it's posted in this week's class. So if you come to this one, you can actually mouse over, all these different areas, and it will give you a view of specific components so that you can read about the involvement of different organs within the immune system, the lymph system itself, the spleen, the skin. So anything you want to mouse over in this diagram, you can learn just a little bit more about it. So the lymphoid system has three major components. It's got lymphoid organs, it's got the circulatory system, and the lymphatic vessels itself. And so there's three types of lymphoid organs. There's primary, secondary, and tertiary lymphoid organs. Primary lymphoid organs are going to be the site of lymphocyte generation and maturation. Secondary lymphoid organs are where adaptive immune responses are initiated. And the tertiary lymphoid organs have relatively few lymphocytes but can recruit large numbers during inflammatory responses. And you also can look at different views. So you can look at specific components here. The thymus, we've already talked about a little bit, being this bilobed organ here where the production of our T cells is going to take place. Bone marrow containing the stem cells, so site of hematopoiesis. We've talked a lot about this already as well. So you can take a look at different things here. Peyer's patches we haven't talked about much. So the mucosal associated lymphoid tissue are all the tissues that are highly lined with a mucosal layer. We talked about the intestinal tract being lined with the mucosal layer. And Peyer's patches are discrete units of cells. There's about 30 of them in each human, roughly. And these guys are small, like little immunity units inside the small intestine. So it's usually inside the small intestine, around the later stages of the small intestine, around the duodenum. And these guys will have concentrations of lymphocytes, especially B lymphocytes, and these can help recruit and carry out immune responses. There's also high concentrations of innate immune cells as well, such as macrophage and neutrophils as well in these areas. And so this helps to deal with the number of bacteria that the small intestine actually gets exposed to during the digestive processes. And what's on number 3? Number 3, just looking at the skin, we've taken a look at this in quite detail. This is a tertiary lymphoid organ, and so it has relatively few lymphocytes. So this would be a good example of a tertiary lymphoid organ. So it has relatively few lymphocytes, but it can recruit a bunch if it needs to. Okay, so what happens if we get a splinter and we start an immune response here? So the first thing that's going to happen is that the bacteria is going to penetrate the skin and it's going to get introduced into the body. The dendritic cells that are located in the skin will interact with any bacteria that would come from this splinter entering into the skin, and they actually will phagocytize them, so they'll eat them essentially, and they start breaking down the bacteria along with some other proteins called major histocompatibility complex. They actually will start expressing some of the bacterial proteins on the surface of their own cell. So these guys can act as antigen presenting cells. So an antigen is just any foreign substance that is going to cause an immune response or trigger the immune system to react when it enters into the body. So an antigen is going to be basically just any foreign thing that gets presented to the immune system. When our immune system cells from the innate immune response phagocytize the bacteria, so things like neutrophils or this dendritic cell that's shown here, or macrophage, they will actually digest the bacteria, but they don't digest it to completion. They will leave intact specific proteins that are on the surface of the bacterial cell, and they will actually embed these into their plasma membrane using the major histocompatibility complex as an anchor, and they will then present this antigen or this foreign substance to other cells within the lymphatic system, and these other cells, the B cells or the T cells that are going to interact with these antigen presenting cells, will start making antibodies against that presented antigen. So once our bacteria is going to be displayed on top of the dendritic cell is going to leave the skin and it's going to enter into the bloodstream, and it's going to travel to the nearest lymph node to present the antigen. So we'll see it's traveling. Whoops, I went ahead, darn it. So it's going to travel through the bloodstream until it reaches a lymph node. So when the lymphocyte actually enters the lymph node, so two things can happen. So if an antigen that is its complement is presented, it will stay in the node and it will become activated. If not, it's going to continue out of the lymph node. In this case, both T and B cell pathways are activated. So we can see, if you click on these different systems, you can actually see an overview of the activation pathway or response pathway. So we'll click on our T cell over here. So if T cells interact with an antigen presenting cell, they start dividing, and you can see these guys match and start producing more T cells that are going to be able to recognize that antigen. Same with the B cells. Our B cells can interact, they start multiplying, they actually are going to differentiate, and they start expressing soluble antibodies. So these guys are known as IgG predominantly and IgA, and we'll talk about the structure of these soluble antibodies later on, probably in the next talk. So these antibodies on the T cells stay fixed on the T cells. These guys go around and then they recognize the bacteria, and they can link them, bring them to macrophage or other immune system cells that can engulf them and cause phagocytosis. So there's going to be multiple steps from going to a stem cell to the primary B cell. We've already talked a lot about this differentiation process, but it starts at the hematopoietic stem cell again, going through the lymphoid line in this case, and turning into a primary B cell. So primary B cell has antibodies that are associated with it. So they get made by the stem cells, they travel through the circulation, and they end up squeezing out of the capillaries. and they'll end up traveling around the tissue until they reach a lymph node. So once they reach the lymph node acts as a filter for antigens from the lymph. And the lymph node is usually bean-shaped, and the lymph from the local tissues is then going to be transferred by afferent lymphatic vessels to the regional lymph node. So the lymph is just the fluid that surrounds the tissue. So remember we said that the fluid is slowly escaping from the bloodstream or the circulatory system into the surrounding tissues. And so this has to be returned to the circulatory system, or else it would start to pool within the tissues and become a problem. So the lymph nodes actually act as small areas within the lymphatic system that are going to have heightened levels of immune system cells, such as the B cells, and these will be places where if you have an antigen presenting cell like that dendritic cell that comes from the splinter, it's going to travel and it's going to go back to a lymph node where it can meet up with actually one of these B or T cells that is present inside the lymph node. And if it does this, it's going to present antigen to that cell. In this case, it's a B cell, which matures then and starts to produce soluble antibody that is directly complement for the antigen that was presented by the antigen presenting cells. So these products then would leave the lymph system. So once these guys mature, they actually get sent out into the bloodstream where they can produce antibodies and help fight the overall infection that is occurring within the body. So if it's at a specific site, in the case of our splinter, these immune cells would then be recruited to that area. They have the specific antibodies that are specific for the antigen that was presented from the bacterial infection at the site of the wound, and the antibodies that get generated and produced will recognize the bacteria, bind to it, and will cause the macrophage to recognize it easier and engulf it and get rid of it, or other natural killer cells. So this is our process of T cell maturation in this case. Again, going through the circulatory system. Goes through the hearts and the lungs and ends up in the thymus. So remember, maturation of our T cells is occurring predominantly inside the thymus. So there's two major types. There's CD4 and CD8 T cells. So once it gets into the thymus, it has the ability to become either a mature CD4 or a CD8 T cell, and these are determined by the receptors that lie on the surface of this cell. So you can see there's a CD8 marker and there's also a CD4 marker, and then there's some additional proteins that get expressed on here. There's going to be a CD3, and the TCR is the T cell receptor, and this is probably one of the most important components of the T cell. It actually will recognize major histocompatibility complex proteins that are expressed by the antigen presenting cell in conjunction with the bacterial fragment or the other infiltrator or cause of the infection within the body. And so it's expressing this antigen, but it's expressing it in association with this other protein complex that this protein here is going to recognize. And so it helps the T cell recognize the shape of the antigen, and then the T cell can then mature, and in this case it's maturing into a CD8 containing T cell. So the antigen presenting cell binds to the T cell receptor, and you get your mature T cell. This gets sent out now into the bloodstream, into the circulatory system, where it can end up in a lymph node. So once it leaves the thymus, it can enter the general circulation and end up within a lymph node, just like our B cell, where they can interact now with the antigen presenting cells that will cause it to expand. So you have clonal expansion, and once this clonal expansion happens, these cells can go off into the circulatory system where they can fight the infection directly. All right, so one of the landmark experiments that took place in immunology was figuring out how the immune system could make so many different antibodies to millions of different antigens that would be presented to them. So to achieve this diversity needed to protect any type of antigen, the immune system would require millions of different genes then encoding different antibodies. It seemed impossible that our entire genome could house so many genes to create so many different antibodies. So Susumu Tonegawa was a Japanese researcher. He won the Nobel Prize for Physiology and Medicine in 1987, and it was due to his discovery of how these immune cells are actually producing such antibody diversity. And so he discovered that they underwent a vast amount of DNA rearrangement within the regions of the genome that encode for the antibodies. So what he did in his experiment was he compared the DNA of B cells, so mature B cells, so a myeloma cell is an example here, and he compared them with the embryonic stem cells from a mouse. And so if you looked at embryonic cells and then cells that were mature from the adult mouse, what he could show was that in the mature cells that there had been a vast amount of rearrangement within the gene loci that encode for the antibody structure. And in the embryonic cell, there was a lot more diversity left. So it looked like there was actually specific gene rearrangement of the genetic material. So you'd have, as maturation occurs, and these cells get exposed to the antigen presenting cells, they become committed and they rearrange their chromosome, their genetic material, so that they become outfitted to produce antibodies that are specific to a specific antigen. And so our helper cells, or our precursor cells that make the B cells and T cells, are going to have this pluripotent capacity to produce all kinds of different antibodies until they get exposed to a specific antigen. And that's when they actually will start to differentiate, and they will commit to producing a specific antibody through this process of genetic rearrangement or DNA rearrangement. So if you go to this website and you check out our immunoglobulin rearrangement exercise, you can actually see that the splicing occurs between different regions of the genetic material, and at each of these different segments, they correspond to segments of the antibody. So the V domains are the variable regions, and so when we talk a little bit more in a few minutes about the regions of the antibody, we'll see that the pink regions up here are going to be the ones that are going to bind with the antigen. And so these have the most diversity and flexibility within the antibody. There's also these diversity regions, which are in D, the joining regions, which are labeled as J regions, and then the constant regions, which make up the heavy chains of the antibody, or the constant areas that don't have as much diversity. But if we kind of play around with this, we could select different regions of where we would make different splices. So if we want to include the variable regions 1, 2, and 3, we could select here, and we'll include everything upstream of here. If we want to only include one diversity region, we can click on 1. These 4 would then get spliced out. And we'll click here, and then we'll keep these 3 J regions, or joining regions, and then we can select Go, and we will see what our gene is going to actually look like. So these are the types of experiments that he could run. So he could run out genetic material that had been already spliced and rearranged, and then he could use a small probe. So in this case, you've got two probes, one that's over the J3 region and one probe 1, which is up here. And you'll see for our splice variant that probe 1 does not pick up any signal in our DNA, and that is because it has been spliced out of our product. We only contain the first diversity region, which no longer gets recognized by probe 1. Genes for the immune system that are going to produce antibodies are given the name Ig for immunoglobulin. And so these are globular proteins of the immune system, and that's where their name comes from. And one of the most striking features then of the immune system is its remarkable ability to recognize and distinguish between self and non-self, and it also can recognize millions of foreign invaders that come into our body that our body has never been exposed to before. The major cell types that are used for this adaptive immune function that we seem to have to recognize foreign substances are the functions of the B and T cells, and these utilize specific receptors that are going to recognize the foreign antigens. So each individual B or T cell is going to produce a receptor with a single but unique antigen binding specificity. So collectively, the lymphocytes then express an almost infinite repertoire of different antigen receptors. So calculations suggest that there's up to 10 billion variants. So this enables the immune system to respond to almost any incoming antigen. So this genetic rearrangement of our antibody binding regions actually enables this great diversity or vast array of different antigen receptors to be possible. So we'll talk in a little more detail about antibodies, what they actually look like, and the mechanisms that T cells and B cells actually use to create these diversity of receptors. So our immunoglobulin or our antibody has a base structure that looks like this. It's composed of 4 peptide chains. It's got 2 heavy chains, so the heavy chains are longer and heavier. And then it also has 2 light chains that are attached here. And the different peptide regions are held together largely by disulfide bridges between cysteine residues in the amino acid structures. So you can see the presence of these cysteine links as the little black lines in our diagram here. So we can also see that the segments of our antibody are color-coded just the way they were in our last diagram where we were looking at gene rearrangements. And you can see that the blue regions represent constant regions. So these have a lot of homology or similarity between different antibodies. So our blue regions are held fairly constant. And then our pink regions are the regions that are highly variable. And we can see that each antibody molecule is going to have 2 major areas up here that are going to recognize the antigen. And so these guys are duplicates of each other. And so each antibody molecule recognizes 2 antigen binding sites. And you can see that there's the joining regions and the diversity regions are also included up here in the variable regions. So the diversity that does occur within the heavy chain region or the C-terminal portion of the peptides actually helps determine what type of antibody that this is going to be. So the antibodies or the immunoglobulins are divided into 5 major isotypes. You can have the IgM, the IgD, IgG, IgE, and IgA types. And so we'll talk a little bit more detail about these different types. So B cells during their lifetimes can switch between the different constant regions. So this is known as class switching or isotype switching. But we won't go into more detail about this. But a B cell has a propensity to produce IgG for a little while, and then it can switch to producing IgD or IgM or IgA type antibodies. So it has a lot of diversity when it is fighting an infection. However, once it commits to a specific antigen, the variable regions will be selected for, and then those guys cannot be changed anymore. So a single B cell is going to recognize a specific antigen, And even though it can produce different subtypes of the antibody molecules, it'll always be specific for that particular antigen that it got selected for. So prior to Susumu Tonegawa's experiment in 1976, immunologists were very puzzled at how the diversity of antibodies could be generated by our fixed genome. So this possibility of having genetic rearrangement that occurs within the chromosome of the host material to produce all of this genetic diversity can create billions of different epitopes that can recognize foreign compounds that come into the cell. And this is quite a remarkable feature of our genetic material. So if we zoom into the cell and take a closer look at what happens, we'll go to the actual chromosome, and you've got your genetic material here. And within a specific segment, you're going to have the genes that are required to produce the different antibody regions. And so we saw this just a little bit when we looked at the Tonegawa experiment at the beginning of this talk. But you'll see now that this matches up very closely to our regions on our antibody. So there's the variable segment, we have the diversity and joining segments, and then the constant segments of the antibody region. And so this is a diagram for our heavy chain right now. So again, the V region stands for variable, the D the diversity region, the J for the joining region, and the C for the constant region. And so oftentimes you'll see this called VDJ recombination. And so they're referring to the switching or gene swapping of the different exons that exist within these regions. So you can have mixing and matching of these different regions. You can splice out different exons, and you can get different combinations within the variable, diversity, and joining segments of the antibody that are going to give a high diversity to our antibody pool to be able to bind to specific antigens. So if we use the mouse as a model for our IgG genes, so for our Ig genes, our immunoglobulin genes, you can see that there's two different genetic regions for the light chains. There's the lambda light chain region and there's the kappa light chain region. And then there's one area on the chromosome where the heavy chains are actually produced. So the genetic segments encoding heavy chains in the mouse are found on chromosome 12. Within the humans, these would be on chromosome 14. And then again, the light chains can be expressed by two separate loci. And one is found on chromosome 6 in the mouse. This is the kappa locus. This would be found on chromosome 2 in the humans. And the lambda locus on chromosome 16, which would correspond to chromosome 22 in the humans. So note in this figure that the lambda and the kappa loci for the light chains, they lack the diversity segments that are housed within the heavy chains. And we saw that in our antibody figure in a couple of slides ago. So if we just go back and take a look, you can see that this diversity region here, that green part, only occurs within the heavy chains of the antibodies. You don't have the diversity region occurring in the light chains. Okay, so if we jump back forward, whoops. So this VDJ recombination can form a large amount of diversity. And you'll notice some specific features within the Ig genes. There's a leader sequence, and this is shown in white, and you can see them here. And there's multiple leader sequences typically throughout the variable region. The leader sequence is important because it helps direct the transcript once it gets made, so the messenger RNA, to be guided to the endoplasmic reticulum. So these get encoded on ribosomes that are associated with the endoplasmic reticulum, and the antibodies are actually produced and housed within the ER. So they're not cytosolic. They have to get packaged into secretory vesicles so they can be presented either on the surface of the cell or they can be exported out of the cell if they're a soluble antibody. You'll also see some segments that have this psi Greek symbol in them, and what these guys mean is that these are actually pseudogenes. So you can see one here in the J region, one up here in the J region as well. And so what this means is that these have evolved over time but are no longer functional. So they're a defective gene that can't really be translated into a protein anymore. So sometimes that happens over evolution. Evolution is a random process, and so it can make mistakes that are not plausible for the development of a new gene to occur. And so these are kind of the evolutionary mistakes that occur over time. So our leader sequence that is going to help guide our transcript to the endoplasmic reticulum actually gets cleaved off during the peptide synthesis. So you lose that leader sequence once the protein has been expressed and is now centered into the endoplasmic reticulum. So this is something that will not be a final component in the antibody. And then interestingly, with our pseudogenes, if you look at the human chromosomes and human loci, there's actually more pseudogenes that are contained within the human genome than there is with the mouse genome. So this slide is just giving an example of how this rearrangement could occur. So you could have genetic rearrangement. This is not splicing in the messenger RNA. This is actually happening in the chromosomal DNA. You get this genetic rearrangement. And so once a B cell or a T cell goes to maturity and it gets selected for producing a specific antigen, it will actually rearrange the gene sequence, and this will then be housed on the DNA. It will then get transcribed into the messenger RNA, and you can get different splice variants. Once that messenger RNA gets produced, it's going to get targeted to the ribosome that's located on the endoplasmic reticulum, and you'll produce your antibody protein from this. Production of the light chain occurs in a very similar way. You have genetic rearrangement once the cell is committed to recognizing a specific antigen, and then you'll get your primary RNA transcript from this newly designated genomic rearrangement. Then you'll have the RNA splicing. You'll get your final product. This is then translated at the ribosome associated with the endoplasmic reticulum, and you will get the production of your light chain. So once the light and the heavy chains have been produced, they're going to come together in the endoplasmic reticulum and form this heterotetramer. It will bud off from the ER, go into the Golgi, where it'll undergo final processing, get placed into another secretory vesicle, and transferred to the membrane. So if this is a soluble antibody, it'll be released from the cell, go into the bloodstream, where it can seek out and recognize its specific antigen. Other antibodies of different classes and different subtypes could be embedded into the membrane of the cell, and these guys are going to stay on the surface of the cell then and recognize the antigen. So B cells are special in that they can produce both soluble and isolated antibodies that are on the surface of the cell. Within the mouse system, if you're looking at the light chain loci, the kappa or the lambda loci, the mouse actually will try to rearrange the kappa loci first, and if that fails to recognize the antigen properly, it can initiate rearrangement on the lambda locus and then make antibodies that are specific for that antigen in that fashion. So within humans, no such preference for the kappa versus the lambda locus has been described yet. So how is VDJ rearrangement actually controlled? So there's some important keys to this process in the presence of signal sequences that flank each segment. So within these regions that do not encode for the actual regions of the protein, whether they're the variable regions or the joining regions, there's signals that can be housed within this genetic structure in here that help catalyze the rearrangement. And so these require some proteins actually to help out in this process as well. But this is just giving some examples of some of these signal sequences. that are located in between some of these regions. So here you have the joining exon and the variable exon, and then this would be the intron sequence, so the non-coding region. And you can see that there's specific sequences that are shown in here in the orange and the blue color that are going to help with the rearrangement. And so one of these sequences is actually a palindromic heptamer, so it contains 7 bases. And basically, if you look at the sequence going in the 5' direction to 3', you'll see it's CAC AGTG in this case, and you can see that a palindrome contains the exact sequence going back in the reverse direction on the opposite strand. So this strand is going 5' to 3'. This strand is also going 5' to 3', but in the opposite direction. So if we read this in the 5' to 3' direction, we can see it's the same sequence that's going across the top. So it's also CAC, and then this base is different in the middle, but then it's got GTG. So it actually is a 6 base pair palindrome with an N in the middle, so the middle base doesn't have to be the same. But you can see that you've got this repeating sequence motif, and this helps to allow for proteins to actually recognize this sequence and bind at this region so that you can get the rearrangement at the correct place next to the 2 segments that are going to get joined together, the variable region and the joining segment. You'll also have these around the diversity segments that are located on the heavy chain as well. So another interesting feature that you'll note here, so you've got this palindromic heptamer. It's separated from another nonomer sequence that helps for recognition between the different segments. And you can see there's 23 bases between this one, and then there's 12 bases between this one. And interestingly, it's been shown that segments that are flanked by a 12 base pair RSS sequence, so the RSS is the recombination signal sequence, so a 12 base pair recombination signal sequence almost exclusively pairs with the flanked segment that's with a segment that is flanked by a 23 base pair recombination signal sequence. And so this rule is known as 12-23 rule, or 1 turn 2 turn rule. And it makes sure that the order will always be V to D to J in the arrangement. So you can see the variable region is flanked by a 23 in this case, and the D region is flanked by the 12 spacer in the case of the heavy chain. So you've got to have the variable region followed by a diversity region, and then the diversity region can be followed by the joining region in this case. In the case of the light chains do not have the diversity region, and you can see that the 23 flanking regions are going to be found adjoining to the joining regions, whereas in the lambda light chain, the 23 flanking region is around the variable region, and the 12 flanking region is around the joining region. So this just enables the enzymes that are going to join these regions together to have recognition sequences, and then they also have these signal sequences of the bases in the exon between the heptamer and the nonomer so that they can determine which areas to actually splice together, who gets joined with who when you're building this chromosomal backbone. And so some of the proteins that are involved in VDJ recombination or rearrangement include lymphocyte-specific proteins, which are shown up here. One of these is the recombination activation gene, so the RAG protein, and the other one is the TDT terminal deoxynucleotide transferase. And so this is a polymerase that actually can add up to 10 non-template DNA nucleotides to the ends of the VDJ segments. And this is either during or prior to the process of joining. So this guy can add just a little bit of diversity to the segment by adding some extra bases. And so this gives further diversity to the VDJ regions to help really tighten up the antibody-antigen interaction. And then DNA repair enzymes are also involved in the process. And so these guys are called the NHEJ, or non-homologous end joining machinery. So you need these auxiliary proteins to make sure that the rearrangement is completed properly and that all of the amendments to the DNA get done correctly. So some of the features of our DNA repair enzymes, they're actually going to mediate the ligation of the ends of the gene segment. So these guys can mediate the splicing, but then you need to rejoin the backbone of the chromosome together to make sure that the phosphate sugar backbone is replaced and connected again. And so the DNA repair enzymes mediate this function. So two proteins that are involved in this process are the Cu70 and the Cu80 proteins. These proteins form a heterodimer, and they bind to DNA ends, and they recruit proteins called DNA protein kinase Cs below to the site of the damage. So these protein kinases, if you'll remember what a kinase is an enzyme that phosphorylates another protein or another enzyme at the serine or threonine residues or amino acids. And so what this does is the DNA-dependent protein kinase becomes activated when it binds to the DNA ends. And it results in the phosphorylation of the DNA protein kinase itself and another protein that's called Artemis. So Artemis can then open up any hairpins that have formed on the non-coding ends. And then you've got the XRCC4 and DNA ligase 4 in the repair system that are going to finally join the two segments together. So this just makes sure that you don't get folding or weird binding of any DNA molecules that might have been cleaved during this process of recombination, so that you don't have it kind of stacking up on itself and forming these hairpin loops. It'll take any of that secondary structure out so that you can actually join the two ends of the molecule back together. So this is just a diagram of our RAG protein working. So it's mediating the splicing then of these two segments. So you can see it's cleaving at the proper sites, the 12-23 rule. And so this is forming a signal joint. This is the part that's spliced out. It's going to give it to the recombination or the repair proteins and the polymerase, which can then do a little bit of rearrangement, add in some extra nucleotides, and then this guy is going to ligate these guys together to make sure that the DNA is sealed properly at that point. So that's that compilation of repair enzymes that would be used at this process. So that contains that protein kinase and also the Artemis protein. So the diversity in between these regions, in between the joining region and the variable region, there's extra bases that can get added in here by the TDT, terminal deoxynucleotide transferase. So again, this guy can add some extra bases in there. So 10 non-template nucleotides, it's just randomly going to add in some extra bases. And that, again, is giving this extra diversity when this guy is actually going to go in there and join the segments together. So again, we'll just run through this little clip showing the splicing of the gene variant. And again, this is happening in the chromosome. This is not a messenger RNA at this point. The actual chromosome is being rearranged when an antigen binding cell or an antigen presenting cell binds to a B cell or a T cell. These rearrangement processes are going on in the chromosome, and they permanently alter or fix the cell to producing a specific antibody. So we've talked about several different mechanisms that help contribute to antigen receptor diversity. So this is a basic summary of these things. So this is our VDJ rearrangement. So we saw combinatorial diversity. So there's existence of multiple V, D, and J gene segments that can be rearranged in different combinations to form different VDJ combinations or different antibody loci. So there's multiple germline segments, and you have combinatorial joining of these regions, and the combinatorial association of the light and heavy chains as well. So you've got combination of the VDJ regions, not only of the light chain, but also of the heavy chain. And so you can have diversity even within these two segments, so that even if you have like one B cell that's producing the same lambda region of the light chain as another B cell, they might be producing a different variable region within their heavy chain. And so they produce just slightly different antibodies from one another. So you can achieve incredible diversity just by this combinatorial diversity that remains in effect within the development of B and T cells. There's also this junctional diversity then, and so this gets back to that TDT enzyme that actually can mediate the generation of new nucleotides that occur between the joining regions of the variable to the diversity segment, or the diversity segment to the joining segment, or in the case of the light chains, the addition of the variable to the joining regions directly, skipping the diversity regions. So up at the top we talked about the combinatorial diversity. We're just going to slide down so we can see the full junctional diversity and somatic hypermutation as well. So junctional diversity is going to give this flexibility, and so our P nucleotide addition, or random cutting of the hairpin loop, generates P nucleotides, and then you have variations in trimming of the coding ends, and then that nucleotide addition by the TDT protein that introduces more segments. So the P nucleotide addition, or random cutting of the hairpin loop, is going to generate a little bit more diversity as well. So you can see we've got nucleotide addition at this place. So if you have a little bit of a hairpin loop that forms between the DNA ends, this can get cleaved in different ways to leave different amounts of overhangs, so that when you actually join these guys together, that they actually can fill in different lengths. And then that TDT enzyme is going to add, in addition, that TDT enzyme is going to add up to 10 new bases in between here as well. So you get functional diversity between that little linker region between the two major exons that get joined together. So this is going to add junctional diversity between the two major variable region and the joining region domains as they come together. And then there's this process also of somatic hypermutation, which only occurs in the B cells. And this is something we haven't talked about yet. But normally when DNA polymerase works to replicate the genomic DNA, making sure that it does so correctly is very important for maintaining the integrity of the genome. And so typically DNA polymerases only make a mistake in about every million bases. So 1 in a million bases will get altered or changed. But you can see that within this B cell region of the genome, so within either the lambda region, the kappa region, or in the heavy chain gene region, you can see that there's a heightened propensity for mutations to occur within these regions. So they've increased from about 1 in a million to 1 in 1,000 per cell division. And these cells usually are rapidly dividing if they are going to be instigated to help alleviate an infection. They get activated, called into service if their antibodies recognize the antigen, or if they've actually docked with the antigen presenting cell, they'll get activated to produce the antibodies that will recognize that specific antigen. And so when this happens, they're rapidly replicating, and you can see that there's a heightened propensity to make mutations within these areas. This is going to increase the random diversity within the antibody as well. And so this can increase the amount of the selectiveness of that antibody for the actual antigen. So you can make small modifications by inserting just random mutations to help the antibodies evolve over time to bind better to the antigen. It's really an ingenious method. And so I think we'll stop here for our antigen recognition segment, and we'll go to looking at how antigens are presented by major histocompatibility complexes on the antigen presenting cells.